Okay, so let's get started. Okay, so hello everyone. Welcome to today's ImmuZoom seminar. And it's really my great pleasure to welcome today's speaker, Dr. Axel Kallies from Melbourne, Australia. Just a very, very brief introduction about Axel. Axel currently is a professor for molecular immunology at the University of Melbourne, Australia. He completed his PhD in Berlin, Germany in 2002. And after a postdoc training, he established his lab and then started his own independent research program at the Walter and Elsa Hall Institute in Melbourne, Australia. In 2017, he was recruited to the Department for Microbiology and Immunology at the Peter Dorsey Institute at the University of Melbourne. His team studies the molecular and the metabolomic control of lymphocyte differentiation in response to antigen. The main interest of the lab is to understand the molecular pathways which require for CD8 T cells and regulatory T cell differentiation. So his lab has really developed and applied different genetic and molecular approaches to T cell biology, which include novel knockout and reported mouse strains, metabolic techniques, transcriptional profiling, chromatin immunoprecipitation, and accessibility chromatin sequencing. So Axel's lab really published a lot of key studies in high-profile journals, which detailed the roles of transcription factors and cytokines in differentiation, clonal expansion, and cellular metabolism of lymphocytes. Over the past few years, his lab has developed a focus on T cells in response to chronic infection and cancer. His lab mainly covered different aspects of the research, which including development of effector regulatory T cells, sex-specific regulation of depocyte tissue immune cells, precursor exhausted T cells in chronic infection and tumors, and also tissue-resident memory T cells. So I think without further ado, we are very much looking forward to your talk. Axel, thank you so much for doing this for us.
Well, thank you so much for this introduction, and thank you very much for the invitation to speak here. It's actually a great honor, and I was really looking forward to this presentation and meeting you guys, even if it's just on Zoom. But yeah, great to be here. I guess I will start sharing my screen. Hopefully that works. Where are we? All right. Somehow I can't. I'm not seeing my. Can you see my screen?
Yes, I can. All right? Perfect.
Okay.
Yeah.
Good. Well, as I said, I'm very honored to be here and speak to you today about T cells. So I got to focus on cytotoxic T cells or CD8 T cells, specifically in chronic infection and cancer. That's where my lab has focused on over the last few years. So we still, as we heard in the introduction, have a program on regulatory T cells, which is also largely linked to Tregs in non-lymphoid tissues, including tumors. But in the interest of sort of focus and time, I will speak about our CD8 T cell work today. Something's not working. Okay, now maybe. Okay, as I said, my lab is particularly interested in the molecular control of T cell differentiation. Oh, this is going way too fast here. And as you know, T cells in response to antigen will start proliferating, will give rise to effector T cells that have a specific transcriptional repertoire, such as Tibet or BLIMP expression, for example, quite famous transcription factors that most of you will know. We will also get memory T cell development in response to antigen presentation and proliferation. And these memory T cells differ from effector T cells, of course, by their transcriptional profile, again, driven by specific transcription factors such as TCF1 or EOMS. If you have antigen that persists, such in chronic infection or in tumors, T cells can exhaust, and this is going to be the main topic of my talk today. And exhaustion, as most of you will know, is again driven by a set of transcription factors that directly respond to TCR stimulation. So that is largely factors such as NFET, specifically NFET-C1, R4, but F belonging to the same group of transcription factors that are driven by antigen. And these factors that induce a transcription factor called TOX. And TOX is really quite prominent these days as a specific factor that is upregulated in exhausted T cells. And all of these questions, of course, are very interesting from a clinical point of view, because as you know, exhausted T cells can be to some degree invigorated by immunotherapy, specifically PD-1 blocking antibodies, very successful in the clinics in driving the regression of some cancers, at least in some patients. And that's why I guess studying exhaustion and maintenance of exhausted T cells, response of exhausted T cells is very important. So we are doing a lot of work with LCMV, lymphocytic choriomeningitis virus, which we really use as a model to study T cell function and exhaustion. And again, it's a very famous model that a lot of labs use, and most of you will be quite familiar with it. But just to bring you up to speed, we have fundamentally two different ways of looking at LCMV infections. One would be the acute LCMV Armstrong infection, which would be cleared within 1 week. There's other strains that are also acute, for example, and all of these strains are cleared within a few days, and you get a typical acute T cell response that in the CD8 T cell compartment gives you, let me get my laser pointer, gives you acutely stimulated T cells that make a lot of cytokines, granzymes, of course, interferon gamma, TNF. However, if you use different strains of LCMV, for example, 13 or docile, these infections can persist for many months or even lifelong, and you get the phenomena of exhaustion, which I mentioned already, which is really a loss of function, in particular a loss of TNF and a downregulation of interferon gamma production that is very obvious in these infections. So coming back to the classical model of T cell differentiation that most of you will be familiar with, we have naive T cells that have a certain repertoire, a transcriptional repertoire that is partially driven at least by this transcription factor TCF1. Once these guys see antigen, they will proliferate, and as we discussed already, give rise to effector cells that now express molecules such as BLyMP or TBET and downregulate TBET. So these guys make cytokines, make cytotoxic molecules such as granzyme. And the textbooks will tell you that some of these cells will then sort of be left behind after the antigen is cleared and develop into memory T cells that again express TCF. And the other aspect that textbooks will tell you that memory T cells don't really develop in the context of chronic antigen, but as we already mentioned, exhausted T cells differentiate. And these cells not only lose function, but they also express a whole suite of inhibitory receptors, most famously PD-1, which is really a hallmark of chronically stimulated T cells and the targets of immunotherapy, as you know. And TIM3 is another one that is studied a lot and is really upregulated on exhausted T cells. So that's the sort of truth that was in textbooks until about 5 or 6 years ago. But then this whole picture changed, and it's quite important to realize how dramatically the understanding of chronic T cell responses has changed over the last few years. So what happened here was that a whole suite of papers was published in 2016, one of them from our lab, but others from very prominent labs like Rafi Ahmed's here in Nature, for example. And what these papers collectively have shown is that the situation in chronic antigen stimulation is quite a bit more complicated. And in reality, exhausted T cells are actually heterogeneous and consist of what we now call precursors of exhausted T cells and fully exhausted T cells. Both of these populations express PD-1, but differ, for example, by expression of certain molecules such as Ly108 or TIM3. And the interesting aspect here really is that these precursors of exhausted T cells, they express TCF1, and they have the ability to self-renew over long periods of time. And as the name suggests, they can give rise to these fully exhausted T cells. So they really act as progenitors or precursors of the terminal differentiated cells. And even more interesting and clinically important, of course, is that collectively the data show that it's really only this precursor population of exhausted T cells that responds to checkpoint inhibition, because these guys really don't have any potential for proliferation. and further differentiation. So it's really that population. And that really sort of invigorated the whole field and really got my lab very interested in understanding this slightly more complex relationship between these different cell types. So what we and many others did at the time was to do transcription profiling. Again, we used the chronic LCMV model that we already discussed and simply compared precursor cells with terminally exhausted cells sorted based on surface markers, and looked at transcription factors and other molecules that are differentially expressed. And as you see here, these cells are quite different. There's hundreds, or depending on how you look at it, even thousands of genes that are different between these two populations, despite the fact that they all express PD-1, despite the fact that they're all chronically stimulated. And there's many transcription factors that are specifically expressed in one or the other population. And we heard already about TCF1, which is transcribed by TCF7. And another factor that we found at this time was ID3. And ID3, again, is a transcriptional regulator that is quite famous in the T-cell field because it's the counterpart of ID2, which is another regulator. So ID3 is actually one of these molecules that is known to regulate different fate in the T-cell field. So we ended up getting an ID3 GFP reporter in order to look at this a little bit more carefully. And what we found is that ID3 is really a perfect marker for these precursor cells. So if you just look at antigen-specific CD8 T cells in chronic infection, in the chronic state of the infection, you can see that all of the antigen-specific cells are PD-1 positive, but there's a very distinct population of ID3 expressing cells that we then characterized as these precursors. They down-regulate or never really express TIM3, so they're negative for TIM3, and they really co-express TCF1, which is, as we discussed, the main factor that drives precursor fate in exhausted T cells. So ID3 is really specifically expressed in precursors, and in some ways, which I don't have time to go into, a better marker than TCF in a broader spectrum of diseases. So, and just to give you a little idea how this sort of looks like from a functional point of view, we tested the precursor function based on ID3 in this very simple model here. So what we did is we infected mice that contained TCR and transgenic cells that express ID3 GFP, and then sorted, after establishing the chronic infection, we sorted ID3 positive cells or ID3 negative cells and transferred them into infection matched hosts and analyzed 7 days later. And I'm showing this because we use this technique quite a lot. And what we can see here is that if you transfer ID3 positive cells, you get a decent population of cells that you can identify here by a congenic marker and CD8, so it's about 2%. Transfer of ID3 negative cells is actually quite poor, the outcome, so you get very few cells that survive it, and you can see that the positive cells give rise to both populations, ID3 single positives, which shows you the self-renewal capacity of these cells, and the precursor function because they give rise to these TIM3 positive effector cells, as we see here. The ID3 negative cells don't really do that. They can only stay in this effector stage, and they do it poorly over time. And if you come back a bit later, you don't really find any of them. So really sort of driving home the point that these guys have self-renewal capacity and long-term population potential. So as I said, we were really interested in understanding the system a little bit more, and I show you a few of our older data here. What we did is we tried to see what is really different between chronic and acute infection. And we look at this very early here, day 5. This is really before any major differences occurs on both situations. There's antigen present. But the main difference, of course, is that there's a lot more antigen in chronic infections. So you actually come in with a higher infectious dose. You have really a very high PFU here compared to the acute system. And what you see is that TCF1 positive cells are really already overrepresented in this chronic, developing chronic infection as opposed to the acute stage. And that was a surprise to us because we really expected that antigen would be driving the effector population rather than these TCF positive cells. So we looked at this a little more closely and simply titrated LCMV dose size. So we didn't even change the strain anymore, but just titrated the infectious dose down. And what you can see is that if you go down with your viral titer or with the virus amount that you inject into these mice, you get at the same time a much smaller population compared to the situation where you have developing chronic infection. And even if you just sort of plot the number or the percentage of TCF cells, TCF positive cells with the plaque-forming units in the kidney, for example, you see that there's a very nice correlation. The more antigen, the more TCF1 positive cells you get. Really surprising to us at the time. So that really suggested to us that TPEX differentiation is really initiated early after infection. That was the first surprise. And that really high amounts of antigen-derived TPEX differentiation, again, the opposite of what we expected. So just to give you a little bit more of an idea what these precursors look like. So we, as I mentioned already, performed RNA-seq in multiple different ways and did different analysis, for example, gene set enrichment analysis. And what we found, for example, one of the major pathways, and others found that too, are metabolic pathways that are really differentially enriched between these precursor populations and the effector population. And mitochondrial matrix, for example, one of these pathways that was very strongly enriched among the precursor population caught our attention. And just to give you some very simple data here, so we looked at mitochondrial function here by looking at the polarization and the amount of mitochondria that are present in these cells. And you can see that ID3 positive cells really have a very different profile of the mitochondria compared to the effector population. They really have very strongly polarized and a large amount of mitochondria in these cells compared to the effector cells. So that's quantified here. And even if you image that precursor population, looks very different from the effector population. And that has, of course, functional consequences as shown here. So we did seahorse assay to look at metabolic flux, and you can see oxygen consumption, for example, here over time. And the precursor population in black really behaves quite differently. If you stimulate oxygen consumption, they have really capacity to ramp up their metabolic response here. And this particular ramping up is called spare respiratory capacity. And you can see that the black cells here, the precursor cells, have actually a lot of capacity to respond to stimulus, while the effector population here, these white cells, really can barely respond to this challenge. So that really shows you that they have more oxygen consumption, more spare respiratory capacity, but the utilization of glucose, for example, is quite similar. So the extracellular acidification rate is very similar. So that just shows you how different these cells are in transcriptional profiles, but also in metabolic profiles. So, but of course, all of this just posed more questions, as it should be. So we really became more and more interested in these cells and tried to understand how is self-renewal and multipotency of these cells maintained. So these chronic infections, as I mentioned, can go on for months if you let them. And we have done that. So we looked at these infections after 3 or 4 or 5 months, and you can always find this repertoire of precursors and effectors present. How is that maintained? How, of course, is it regulated that these, or how does it come that these cells mediate response to checkpoint inhibition? What is special about them? All of these cells express PD-1, but only the precursors respond. And how is this really balanced that you have long-term maintenance, but also impaired function at the same time? How does it work? And in order to understand that a little better, we did what many others do. We did more transcriptional profiling. This time we used single-cell RNA sequencing. And the trick that we used here is we actually focused our starting material on precursors. So we didn't just look at the total population of antigen-responsive cells, but we actually sorted specifically using a few markers and ID3 cells that were precursor phenotype, so that we could actually get our handle better on these cells. And that worked quite well. So we have about a third or maybe 40% of our cells in this analysis are precursor cells identified by expression of high levels of TCF7. So these are the precursors. And then we have actually 2 different populations of effector cells that can arise from these precursors. Both of them express granzyme B. That's a sort of hallmark of these TX cells. But as others have shown, there's 2 populations which we also found, one expressing CX3-CR1, which have actually higher effector function, so they are more effector-like T cells, and then we have really useless cells, if you want so, that are down here, that are terminally differentiated, express very little at all of cytotoxic molecules. So that was very reassuring to see, and you can see how very nicely these different populations separate from each other, so very distinct once more. And again, we have self-renewal potency in these cells here, and we really focused our further analysis on this particular part of the populations. What caught our attention when we did pseudotime and RNA velocity analysis, that all of the trajectories that we looked at actually started from this little knob or this little specific population here within the precursor population. And regardless whether we looked at the effector population or the terminal differentiated cells, all of these cells seem to finally originally come from this particular population here. And these cells specifically expressed, for example, CD62L, which is typically a marker of central memory or even naive T cells. And we were slightly concerned initially that this might be a contamination, but it turned out to be not to be the case, because we actually used P14 cells in all of this, which are, of course, all antigen specific and have all seen antigen, and I hope I can convince you that's the case. So what we then looked at, of course, was in flow cytometry, what does this population look like? And once more, this is P14 cells, all of them antigen specific, and this is well and truly into the chronic phase of the response. And if you look at antigen-specific cells here, all of them express PD-1, and there's a distinct population that expresses CD62L. You can also use Ly108, which is a marker for the precursor population, and you get this beautiful little population of TCF-positive precursors. And again, you get CD62L positive cells that are a part of this population. And this is initially a fairly decent population of about 30%. That goes later down to about 10%, but then becomes very stable and stays around for a long period of time at this level of about 10% of the precursors. So it's a small population. Among the precursors, about 10% at later stages express CD62L. So we looked at these cells a bit more closely and labeled them here as TPEX1 and TPEX2, and then we have proliferating cells and perhaps an intermediate stage, different effector populations, and see all of them are transcriptionally distinct. These guys obviously closer to each other than other populations, but again, quite distinct from a bioinformatical point of view. And interestingly, as I mentioned already, CD62L positive cells here in pink, they really show full signs of activation and all the qualities of exhausted T cells. They express PD-1, they express TOX, they express CD44, TOX being specific to exhausted cells, PD-1 perhaps a little lower than the other cells, but well and truly above naive T cells as shown here. We also looked at signs of TCR signaling. So NER77, for example, is a downstream of TCR signaling. And you can see that if anything, these guys are actually higher than some of the other populations for ongoing TCR signaling, even here, or specifically here at later time points, again, a sign that these cells are fully activated all the time. We looked at many other features of activation. So these cells are fully activated throughout the whole process. And as I mentioned to you, we observed that the lineage trajectories seem to all start from this point. And in order to test this experimentally, we went back to this adoptive transfer system that I showed you earlier. In this case, we transferred P14 cells into mice that were infected then with LCMV docile, chronic infection. And after 4 weeks, we came back and sorted the different subsets of CD8 cells that I mentioned to you. So when we do that and transfer into infection match hosts and come back 21 days, so that's 4 weeks plus 3 weeks later, and look at the outcome of all of this, you can see that it's really the CD62L positive precursor cells that have the highest repopulation potential. So there's a nice population here. You really quite struggle to get anything from the other populations. That's quantitated here. Log scale, so quite a distinct difference. So they really are good in expanding and repopulating the host. And they can self-renew, as shown here. So we can still find 62L positive cells, Ly108 positive cells, precursor population, our 62L positive population, and effector cells. Whereas if you just transfer effector cells, they remain effectors. If you transfer these guys, they can go down this track, but can't go back to being 62L positive. So that really suggested that they have the highest capacity for self-renewal and expansion. So how does this all work, and is it important? So we looked into our RNA sequencing a bit more and found that one particular transcription factor, c-Myb, was specifically enriched to some degree, at least in this CD62L positive precursor population. So you can see it here, this enrichment plot. It's also expressed in other precursors, but there's more of these guys here. So we got a MIB-GFP reporter mouse and looked at that in our chronic infection model. And as you can see, naive T cells don't express really much MIB at all. There's very little expression you can find generally in the T cell compartment. But if you infect them chronically and look at antigen-specific cells, you can see that CD62L positive cells within the antigen-specific compartment are actually very high and very distinctly positive for MIB-GFP. And that's quantitated here over longer periods. So initially, very high expression goes down a little bit over time, but specifically the CD62L positive population maintains a very high level of MIB over long periods of time. So of course, that caused us to ask the question, what is MIB doing in T cell exhaustion? Or what is it doing in CD8 T cells in general? There's a bit of literature, but not very clear. So we looked at this with a fresh pair of eyes and got knockout mice and either infected them with LCMV Armstrong, causing acute infection, or docile, causing a chronic infection. And that's a T cell specific knockout. And what you see here after acute infection, not much actually happens. So the mice are happy and healthy, they survive, they have very similar numbers of antigen-specific cells, early, slight decrease perhaps later, but not very dramatic. They develop very normal looking CD8 T cell responses in terms of cytokines and so forth. I'm not showing you that. Memory development, very similar in both situations. So rather boring to start with. However, if you use these mice and chronically infect them, the situation is quite different. So essentially, all of these mice die around day 10 or even a bit earlier than day 10. Very few of them survive, and they all have fairly substantial immunopathology, severe infiltration of T cells in the lung, liver, and surprisingly actually smaller spleens, which is actually a sign of immunopathology in this particular system. And if you look at the CD8 compartment, you can see quite a dramatic expansion of antigen-specific cells in the knockout, as shown here at the acute time point, day 8 after infection. And the survival of these mice is really dependent on CD8 cells. So if we take these chronically infected mice and deplete the CD8 cells, these mice will survive, suggesting that CD8 cells actually cause and mediate the disease and death of these mice after infection. So what is going on apart from numbers? So we looked, of course, at function. And again, quite surprising to us was the observation that in the absence of MIB, cells don't seem to really show any exhaustion. So looking at interferon gamma and TNF, I showed you earlier, so interferon gamma is quite low. TNF is essentially absent from wild type cells that are in chronic infection. But if you look at the knockout, there's heaps of interferon gamma, lots of TNF, really almost looking like an acute infection. So you not only have more T cells, but also more cytokine. Quantitation is here. And also more granzyme, which I'm not showing you as a fax plot, but within the effector population, more granzyme. So that really suggested that MIB is one of the key drivers that actually is mediating functional exhaustion as measured by cytokine production, shown here. So, of course, our question was also, how do these precursor populations look like in the absence of MIB? And the idea was, of course, that it might be important for CD62L positive cells. So we once more infected mice, but in this case, we used mixed bone marrow chimeras where we diluted, if you want so, the MIB knockout with wild type in order to have mice that can actually survive over long periods of time. So we can now analyze at a late time point after chronic infection. And if we do that, these mice don't die. And you find that precursor cells, as identified by Ly108 here, can develop in both situations, but CD62L positive cells are essentially absent from the knockout when you don't have MIB. And that really suggested once more that these CD62L positive precursors are quite a distinct population that is very specifically dependent on MIB. And what happens to the total T cell response if you look over these longer periods of time? So this is a quantification over time of antigen-specific cells. And you can see after the initial expansion, you get a fairly steady number of cells in the wild type. But if you look at the MIB knockout, you get this really quite dramatic expansion of CD8 antigen-specific cells initially, in the early phase of the response. But then there's a crash at around 2 weeks, it starts to really bring down the numbers of cells, and then eventually even to go down below the numbers of wild-type cells that you can see here. And that's really paralleled by a striking loss of proliferative capacity in later time points within the knockout compartment, and that's quantitated here. So initially you get lots of proliferation, and then this really breaks down to hardly anything at later time points, suggesting that MIB really regulates the proliferative capacity of these cells over time. So what are the signals that induce MIB? So as we discussed already, TCR signals are really important for driving precursor cell fate. So we looked at this in a couple of different ways. So we once more compare docile chronic infection with Armstrong infection quite early in the response, when there's still an effector response going that is rather similar to some degree in both. And you can see once more that precursor cells develop in large numbers in the docile infection, and once more you have 62L positive cells in Armstrong to really lack this population. Both of them are very strongly down. You have hardly any 62L positive cells, whichever way you look at it. And MIB-GFP is really much higher expressed in these strongly stimulated cells in chronic stimulation. You get some expression in Armstrong, but a lot higher the expression in chronically stimulated T cells. And if we break that to a much simpler system, this is just in vitro looking at CD8 T cells from a wild-type mouse or a GFP reporter. You can simply titrate the amount of antigen or antigen stimulation by using anti-CD3 at higher or lower amounts, and you can see that driving up the MIB signal here by high amounts of anti-CD3 stimulation is very obvious here. So it looks like TCR signals really are key to driving MIB expression. So what is MIB doing? Of course, once more, we looked at this by RNA sequencing, this time bulk RNA sequencing. So we sorted from these chimeric mice wild-type cells from the wild-type compartment, or knockout cells from the knockout compartment, and we sorted all these different subsets that we discovered and love, and did bulk RNA sequencing. And what you can see here is, for example, this three-dimensional plot where we look at the relationship of these populations or similarity of these populations. You can see, for example, the 62L negative precursor population and the 62L positive precursor population. They are clearly separate, but relatively similar to each other. But if we knock out MIB, then the precursor population looks quite different. There's, of course, no 62L positive, so all of them are sitting here together. And they become quite similar to the effector populations, either from the wild type or the MIB knockout. So quite a dramatic shift in the whole profile of these precursor cells in the absence of MIB. And without going into too much detail, there's a large number of genes that are differentially expressed, of course, between wild type and knockout precursors. And some of them we validated and also think are functionally important. For example, c-KIT. Some of you might know it is of course, a factor that is quite relevant to the hematopoietic system and is usually expressed on hematopoietic stem cells or progenitors in that field. And we could show that KIT is really regulated directly by MIB binding using chip sequencing here and ATAC sequencing to show that it's an open chromatin. And if you look at MIB knockout precursor cells, we found actually that not only they lack CD62L positive cells, but they actually acquire a population of c-KIT positive cells, which is not really that distinct in the wild type. And I won't show you the data, but we have some data now that suggests that KIT actually is directly involved in the proliferative capacity of these precursor cells. So it's not just a marker, but it's also functionally important for the cells. So now coming to checkpoint inhibitor therapy, is any of that important in that context? And we believe it is. So what we did here is we once more performed this sorting and transfer experiment that I talked about earlier. So we took infection-matched hosts and transferred these different populations into these hosts. And this time we now treated also with anti-PD-L1 to see which of the cells can respond to checkpoint blockade. And as you might have gathered, so it's really the 62L positive precursor cells that almost exclusively seem to mediate checkpoint inhibitor therapy. So you get a really lovely expansion of these cells in this particular setup here, whereas the 62L positive negative precursors, which are the dominant clone or the dominant population, don't really respond very well at all, and the effector cells don't respond at all. And that's like the quantification here, once more log scale, so quite a nice difference between the response between these populations. And of course, that also suggests that MYB is involved somehow, and that's indeed the case. So what we did here is we used these chimeric mice, once more well-controlled system, at a late point of the infection, and treated these mice with checkpoint blockade and look at the wild type CD8 population or the knockout population within the same mice. And as you can see, after a blockade of PD-1 signaling, you get this nice expansion of PD-1 positive cells from about 20% to about 35 or 40%. Whereas here, really, in the absence of MYB, none of that happens, really showing you that MYB is really important for the proliferative response in response to PD-1 blockade, and that's quantitated here. So that brings us to the slightly more complicated universe of exhausted T cells that I would like to show you once more here. So we start off essentially with naive T cells that express TCF1 and ID3. And when you give them an antigen signal that is strong and sort of suggestive of a chronic infection, what happens first is that you get really a differentiation of these stem-like exhausted T cells that express CD62L together with PD-1, c-Myb, TCF, ID3. All of this is expressed in these cells. And you get a first wave of effector cells that I haven't really talked about, but these cells are essentially normal effector cells that die within a few days after the infection. Whereas these guys have the self-renewal capacity that we discussed and can then give rise to what we have looked at so far, these normal precursors of exhausted cells that have a certain capacity to self-renew for some period. And these cells now express c-KIT and lose CD62L, and can give rise to effector-like cells that express CX3, CR1, for example, all these terminally exhausted cells. And what happens in checkpoint inhibitor therapy is essentially that you block PD-1 signaling, in particular, importantly, on these cells here, which then induces fundamentally a wave of differentiation that results in more precursors and more effector cells. And all of this, in the end, results in an overall expansion of the CD8 population, of the antigen-specific population, in particular of these effector-like cells that are mediating the response within tumors. So that's the summary of what I just showed you. Most of this is published in a paper that came recently out, so I might not go into much further detail, but maybe in the remainder of the time, do we still have time? I might show you a little bit what we are doing currently. And this is really just sort of trying to expand the model and the findings that I talked about into perhaps clinically more relevant questions and into more complicated models. So one of the questions, of course, is how does this all work outside of the model of LCMV, and is this relevant in tumors, for example, relevant in non-lymphoid tissues? And that's fundamentally what we are trying to address at the moment. And I show you just a little bit of data because it's all ongoing work, and we can discuss it perhaps in more detail another time. But just to bring you back on speed here, so what we are doing, of course, like many others, is we look at anti-tumor responses and T-cell exhaustion within cancer. And one of the models that we're using is 83, breast cancer model, where we use anti-PD-1 in order to suppress tumor growth or let them grow without treatment. And this is what it looks like. And of course, you get, if everything goes well, you get a nice response, tumor shrink, tumor weight goes down. And allowing us to look at all of these questions in the context of tumors. And one of the observations that we made here, and others have shown similar, of course, is if you look at untreated tumor-infiltrating CD8 T cells, so these are CD8 T cells once more in untreated tumors, you find TCF-positive cells, so precursor-like cells, as I described to you, that we see in chronic infection. You find effector cells that express some level of TIM3. And if you come in with checkpoint inhibition, it looks like as if these TCF-positive cells start differentiating and give rise to TIM3 positive cells. That's at least the interpretation that is currently out there. These guys get induced to differentiate into effector cells, essentially dropping in the percentage here, and these guys go up, mediating exactly this therapeutic response. So that's what we also see. But we kind of are interested in understanding a little better what this means and how does this look like compared to a steadily growing tumor, what kind of precursors are in these tumors, what kind of precursors are perhaps in draining lymph nodes, and is checkpoint inhibition really initiated within tumors or perhaps in the lymph nodes or both? So that sort of brings us once more back to the question, how are these precursor cells regulated? What do they look like in the context of cancer? And to look at this in a slightly different manner, we generated another mouse model. And that's really based on an older paper that we published together with Laura McKay quite a few years ago, where we showed that the transcription factor Hobit is very specifically expressed in tissue-resident T cells. And I won't show you the data, but in this paper, we showed that this factor is really specifically upregulated and involved in tissue residency of CD8 T cells. So what we did is we made a tomato reporter mouse that leaves actually intact the Hobit locus, inserting tomato here, and also a Cre recombinase that allows us to manipulate the cells in various ways or delete genes of interest, of course, in tissue-resident T cells. And that mouse works quite well. So this is just a control. So we used these Hobit-Tomato mice infected with acute LCMV, just giving normal memory cells, and looked at a number of non-lymphoid tissues, such as the salivary gland, small tissue, liver, and so forth, and also at the spleen. And what you can see, if you look at CD69 positive cells at this memory time point, that's what you usually use to identify tissue-resident T cells. You can see there's a beautiful population. In the salivary glands, for example, essentially all of them express Hobit. Small intestine, a bit more heterogeneity, but a large population of Hobit-positive cells. Liver, again, nice population. Spleen, as expected, mainly circulating memory cells and only very few cells that express tissue residency markers. So that seems all in order. And in order to now sort of look at tumors in a bit more sophisticated way, we decided to cross these Hobit-Tomato mice with these ID3 reporter mice. So these guys reporting for precursor function and these reporting for residency function. And the idea is that we can do that independently of cell surface markers. And that gives us quite a nice picture. So as you can see here, looking at these tumors and looking at the tumor infiltrating CD8 cells, we have a very nice population of ID3 expressing cells, about 40% perhaps that are ID3 positive. And perhaps a similar percentage of cells express Hobit. And interestingly, about half of these cells express both markers, ID3, GFP, and Hobit-Tomato, suggesting that there is something quite interesting going on. And our sort of initial hypothesis was that all of these cells might represent precursors, and perhaps these precursors that express tomato might be resident in some fact. And we are at the moment looking at various ways of testing this model and looking into this a little bit more. And yeah, of course, and then you would have perhaps normal tissue resident T cells, which have been shown to be important for tumor control. So they might be sitting down here, and maybe classical effector cells or fully exhausted cells down here in this black quadrant. So just showing you once more the dramatic impact of checkpoint blockade. So we used these double reporter mice, which have this picture here, and treated them with anti-PD-L1. And what you can see is quite a dramatic response, as already seen by TCF staining. You essentially induce at least the relative loss of the majority of these precursor cells that are here highlighted by ID3. You also induce loss of most of the tissue resident cells, and you really lose very much the double positive population, perhaps suggesting that these double positive cells are particularly important in the response to checkpoint inhibition. So really highlighting that this whole intratumoral T cell network is dramatically reorganized after checkpoint inhibition, and that's essentially what we're trying to understand now. Further to this point, of course, there's a fair bit of literature that suggests that lymph nodes are very distinctly involved in tumor control, of course, and also in response to checkpoint inhibition. And there's a number of different models out there. So there's some indication that, of course, precursor cells might reside in the lymph nodes. There's also literature out there that suggests that tissue-resident memory cells can reside in lymph nodes and play a particularly important role in tumor control. And other people have involved just classical memory cells in the response to checkpoint inhibition. So there's a few different ideas out there how lymph nodes are involved, and we think we can maybe contribute with our model to that as well. So we looked into this a little bit more closely, and what you can see, and again, it's just a snippet of data that we have. So looking at the tumor, again, CD8 T cells infiltrating the tumor, most of them express PD-1, as you would expect. Essentially, all of them are CD44 positive. The draining lymph node, quite a different situation. You have a very small population of PD-1 positive cells in the draining lymph node, and almost all of these cells have a precursor phenotype, as measured here by Ly108, and absence of TIM3. Whereas in the tumor, you have really a large number of effector cells, but a relatively small number of precursor cells, as shown here. And if you look at CD62L, the marker that we think is important for the most stem-like cells, you can see that in the lymph node, these cells are quite distinctly represented here by about half of them, half of the precursors expressing CD62L versus a very small number within the tumor. So that suggests to us that perhaps the tumor-draining lymph nodes are indeed the ones that harbor an important population of precursor cells. And this is also enforced by this observation here, where we used once more the MYB-GFP mice, this time in context of tumors. And what you can see that in the draining lymph nodes, that's just the wild-type control, you see a really nice expression of MYB-GFP, both among the 62L positives and also to some degree in the 62L negative precursor population. But within the tumor, even although you have some 62L positive precursors, essentially none of these guys express MYB, suggesting that they don't really have that stem-like capacity that perhaps these cells in the lymph node have. And that's a quantification here. So the idea really is to now use all of these models, like the double reporters and the MYB-GFP, and really examine the precursor network within tumors a little bit in more detail, and see where we get in the context of tumor control and actively growing tumors, but also in checkpoint inhibition. And that brings me to the end. So that's a picture of my lab, a few years old already, unfortunately, but most of these guys are still around. And most of the work that I showed you today was done by a really fantastic postdoc in the lab, Carlson Sui. He's been doing most of the MYB-related work. Sharanya has been doing most of the Hobit-ID3-related work that I showed you only a few pictures of, and Sarah has initiated most of the precursor work in my lab. And we are working closely with Daniel Utzschneider and a number of other people. And we have some fantastic collaborators, especially in Germany. So the Buchholz lab in Munich and the Carsten-Müller lab in Würzburg are really important collaborators of mine. And if any of you ever think of coming to Australia, we have beautiful summer weather right here and right now. So it's about 30 degrees outside, sunshine. And we have a fantastic scientific environment, I think, in Melbourne, perhaps not quite the NIH, but if you feel like coming, you just get in contact and let me know. Thank you very much.